Dear students, welcome back to end of year revision, and today we will start with term 1 topic, that is 2.3 DNA, RNA and protein. So first of all, starting with the first KPI, describe the mechanisms by which genetic information flow, and this mechanism is called as central dogma. So how we make the central dogma? It's quite easy. There are 4 major steps you need to learn, which is 2 T's and 2 R's. We will learn those. So first of all, we have DNA, which convert into mRNA, and when this mRNA converts into protein, this is another step. So first step, when DNA converts into mRNA, this is called as transcription, while when the DNA RNA information is converted into protein, this is called as translation. So based on that, our 2 T's are completed. Now we will look for 2 R's. So we know that DNA has the ability to make its own copies, and when this arrow is shown like this from DNA to DNA, this is called as replication. And this replication completes our 1 R. Some of the organisms like viruses, they have their genetic information in the form of the RNA. So when the genetic information is not DNA, it is RNA, so it's converted back to another type of the DNA that's called as cDNA, and then the process goes on. Such process is called as reverse transcription. So we are done with our R's as well. So the 2 T's are transcription and translation, while the 2 R's are replication and reverse transcription. I hope it is very clear. So we will move on to the questions. The genetic material is used as a template for protein synthesis, and we know that genetic material is DNA. Okay, it is used as template for protein synthesis. Which of the following is the correct sequence? So of course, when I said that genetic material, so you must have to start with the DNA. So A is wrong and D is wrong, so B and C can be correct one. But the first process, when the DNA converts into mRNA, it is transcription. So DNA to mRNA to protein, first T, second T, so answer is B. Which of the following is R? So it can be one answer or more than one answer. True for the process shown in the diagram below, and the process that you see in the diagram below is the same central dogma of life. So the best way to answer such questions is to label them quickly, even though labeling doesn't have marks. So this is replication, right? While this is DNA, this is mRNA, and the last one, protein, is already labeled. So this is first T, that is transcription, and that's the next one is second T, that's translation. Okay, so once you have this information with you, you can easily go through the answer. Number 1, proteins are manufactured from mRNA in stage B. So stage B is making mRNA. This is not protein, so this is wrong. Stage A requires tRNA. Stage A is replication, doesn't require tRNA. Stage C represents the process of translation. Yes, we said it is translation, so only C. That is number 3. So only 3 is the correct one. Moving on to the next question. Which of the following processes is carried out by the ribosomes during protein synthesis? So what do ribosomes do? So ribosomes are involved over here. This is the most important stage, that is C, that we study up, translation. So they are involved in translation, because this translation translation takes place on ribosomes, while on the other hand, replication and transcription, these two stages takes place in the nucleus, Okay, while the translation takes place on ribosomes in cytoplasm. So this is good that we have known about the locations of the processes as well. So which of the following process is carried out by the ribosome? Of course, now we know it that this is D, that is translation. Moving to the next. Okay, now we have another KPI. Relate between the structure and functions of the three types of the RNA. So three types of the RNA. So first of all, we need to label them the three types. So make sure you know them. The first one is mRNA. The second one is tRNA. And the third one is rRNA. Now here we have two aspects. Number one, you should be able to understand the structure and function. So the question can be asked in two different ways. Either you identify the structure or you identify the function. So the first thing that talks about the structure, so we have over here, if you see a single stranded, okay, it is single stranded and it's linear, okay, and it has U, that is uracil. Uracil is present instead of thymine, instead of T. So this will be mRNA. So it's single stranded and that we will call as mRNA, and it carries the genetic information from the DNA. This mRNA will go to the rRNAs. That rRNAs make ribosomes, and these are present in the cytoplasm as we said before, and the process of translation takes place over here. We studied in the previous question, and rRNA are associated with the protein to form from the on the ribosomes. And if it is the next one, the last one, tRNA. tRNA is like a loop or a hairpin shape. Okay, it has two ends. On the bottom end you have three spikes, and here you have the code. For example, sorry, not the code, it's anticodon. So here you have anticodon, which is complementary to the codon on the mRNA. So for example, here these three together they are called as codons, okay, while on the other hand these three are called as anticodons. And on the second side here you have a circle like structure, and this is amino acid. So tRNA looks like a hairpin structure, and it has two major labelings. Number one, amino acid, while the second one, it is anticodon. So this is how you identify their structures. Now what is the most important function? So here they transport amino acids to the ribosomes. rRNA make ribosomes, and mRNA carries the information from DNA, which was in the nucleus, to ribosomes, which is in the cytoplasm, for protein synthesis. So mRNA, these are the functions written on the top. Learn as it is. mRNA is long strands of the RNA nucleotides. I told you RNA nucleotide mean they have U, not T, and they are single stranded because they are made complementary to one DNA strand, and this information moves from the nucleus to the cytoplasm. rRNA contain two subunits. So these subunits look like this. From here till here, this is a large subunit, and from here to here, this is a small subunit, and the RNA fits between here in these subunits. Okay, so this is mRNA that fits in between these two subunits. Okay, moving onward, and the rRNA makes up ribosomes. tRNA contain three base pairs at one end, as we discussed already. This is called as anticodon, while the complement that are complementary to the mRNA. Codons on the mRNA, while on the other end they have the amino acid. So they have two roles. Number one, they have the anticodons, and they carry specific amino acids. Okay, moving to the questions. Which of the following is RR mismatch for the type of RNA and its function? So basically we are going to label them with function. So first one, mRNA carries amino acids. No, it should not be mRNA, it should be tRNA. rRNA is a part of the ribosome structures. Yes, that's correct. And tRNA carries genetic information. No, this should be mRNA that carries the genetic information from DNA to the site of the translation. Good. So only uh, which one is mismatch? Oh, okay, over here. Now please be careful that don't go for B, because as I solved it looks like B is the answer, but no, B is not the answer because we are looking for mismatch. So how many mismatch we have? We have number 1 as a mismatch and we have number 3 as a mismatch. So the answer is 1 and 3 only. So this is how most of the students, they get confused over here, even though I could make the mistake when I see only 2 is correct. So I go down and select 2 only. Which of the following is correct match? Okay, now here we need to see the correct match. So it means there are 3 mistakes. Okay, mRNA carries anticodons. No, this is tRNA. Pre-mRNA joins. We will talk about what is pre-RNA and what is pre-mRNA and what is mRNA in the coming KPIs. Pre-mRNA joins amino acids by forming peptide bonds. No, this is basically the mRNA and tRNA role. rRNA contains codons. No, this is mRNA that contains codons. And tRNA carries amino acids to the site of translation. Yes, that's the only one correct over here. Moving to the next now. Write the names of the RNA shown in the diagram. Of course, if it is single-stranded, it is mRNA. So straightforward. If it is a pin shape and you have anticodon over here, this is tRNA. And if it is 2 subunits like these, so this is rRNA. Describe RNA processing. Distinguish between the introns and exons, adding the G cap and its role, and adding the poly A tail and its role. Okay. Now, in this one, the most important thing is that we should know what is RNA processing. RNA processing is a process in which pre-mRNA is converted into your final mRNA. This is also known as post-transcriptional modification, but this is a very long name. I will recommend you better you learn the same as RNA processing. We sometimes also say it as RNA editing. Okay, so we say it as RNA processing or RNA editing. Yes, one more that is called as RNA splicing. So you can choose any name that is good for you for this process in which a pre-mRNA is converted into mRNA. So normally I will go back and tell you how this works. So normally this central dogma, it looks like this. Okay, where I write? Okay, I can write here. So this is DNA. It converts into pre-mRNA. This pre-mRNA converts into mRNA. This pre-mRNA converts into protein. And the DNA can make its own copies. We have all the same steps. So here it is replication. Here it is translation. Oh, sorry, not translation. It is transcription. Okay, and here it is translation. While this one more step that came in between, this one is splicing. This one is splicing. This one is editing. This one is, sorry, the spellings went wrong. Editing, or the one they are using, it is processing. So you can say over here, from here till here, this is RNA processing. Okay, now what happened in this processing? Only 4 major things happen, which are listed on the top. Okay, so what are the 4 major things happening? Number 1, if you see this pre-mRNA, once you have the pre-mRNA, you see that it will get a cap that is a 5' cap, and it will get a tail that is a 3' tail. So 2 editings happen. Now you see here introns and exons, there are the 2 parts. So basically, what are introns? Okay, so introns are non-coding regions which have to be removed. So how you can say it? Introns removed. And the exons are kept. So exons are the coding regions which are kept in your final mRNA. And you get a 5G cap, and you get a poly A tail on the 3 prime. On the 3 prime you get a tail, and on the 5 prime you get a cap, that is a G cap. Okay, so these are the 4 modifications that happen during RNA processing. I will repeat them. Number 1, introns removed. Number 2, exons kept. Number 3, G cap is added on the 5 prime, while A tail is added on the 3 prime. Use the diagram to answer question 7 and 8. So this is the diagram. We have the DNA converts into pre-mRNA transcription, right? So this is transcription, converts into mRNA. This is RNA processing. RNA converts into polypeptide. Polypeptide mean it's a protein. So this is translation. Look, the most important thing is once you are able to read the whole diagram, you can easily answer it. Identify the processes A, B, and C. I already named here A. This is A. B. This is B. C. This is C. Okay, you can put them down. Describe the structural differences between pre-mRNA and mRNA produced after process B. Explain the significance of the difference to protein synthesis. Okay, so first of all, we need to know the changes that happened. Number 1, poly A tail is added and G cap is added. And in addition to that, you see the introns over here, but no more introns down. So introns removed and exons are kept. So it means that these 4 processes, 4 additions you can write down here. Okay, and what else you can see over here? What else difference you can see? Look, pre-mRNA. This is the pre-mRNA, which is quite longer. Okay, and final mRNA is shorter. So you can also say this, they are long, and like pre-mRNA, this is longer and mRNA is shorter. Okay, then exons are kept and introns are removed. Poly A tail is added, and poly G cap is added. So this is how you answer this question. Identify the genetic code and explain how it is read. Genetic code, how you define it? It is a triplet of nucleotides. So when you read the bases as 333, it's a nucleotide. So when you read them as 333, for example, ACG, AUG, or CGA, whatever, when you read them as 333, it's called as a code. And how you read it? You start from the first side, which is over here. You go to the second side and go to the third. That's the last one, and you read them. Whenever you read them, there is a name after it. Follow the name. Please make sure you put, if it is given you as a 3 letters, put the 3 letters in the answer. If it is given you as a full name, put it as a full name. No single letters, no changes as per the table. Okay, so genetic code is a set of triplets. It's also called as codon. It is present in the mRNA and DNA as well, but normally we read it in the mRNA. Each codon codes for one amino acid. There is specific instructions. There is a start codon and stop codons. So start codon is always AUG. Start codon is always AUG. Okay, while the stop codons are UAA, UAG, and UGA. Okay, these are the stop codons. Why we say them as stop codons? Because no amino acid is coded by them. So in the table, whenever you reach to here or here, you write stop, and whenever you go here, here AUG, you say meth. Don't say start. You need to name it because it has a name of the codon that is meth. Meth mean methionine. Okay. Use the data to determine the amino acid chain that would result from the mRNA. So we have an mRNA sequence. Example AUG. So you need to go to the table and you go to A, you go to U, and then come down and you find AUG. So this is met. So same way you will read everything. I'm not going to read all. So this is AUG, it is methionine, GCU alanine, and this one. So you can put them together like this. And UAA, it is a stop. So if comes UAA, you should say stop. Okay. So see, here there is another question. How many nucleotides you have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 nucleotides. Okay, since we know that each 3 makes one codon, so how many codons you have? So simple is that 12 divided by 3, that is 4. So we have 4 codons. And how many amino acids? Each codon code for one amino acid. We read it here. Each codon code for one amino acid. So it means that there is a possibility of 4 amino acids. There is a possibility of 4 amino acids. But since we have here one stop, so how many? So how many stop codons have, how many amino acids have been coded by this sequence? Yes, you can say not 4, it's 3. Why? Because we have one stop codon. But it's not the case in everywhere. They can be different. It depends how it goes. There is another example down. You can just go through it. Which amino acid would be transferred to the position of CUU? So you need to go to the table again. So here it is C, here it is U and U. So this is LEU. Okay, so we will go back to the answer. So LEU, it is leucine. Okay, moving to the next one. Okay, moving to the next one. The chart below shows the nucleotide sequence of P in the mRNA that codes for different amino acids in the proteins. For the nucleotide sequence in the section of mRNA below, answer questions A and B. A. How many total amino acids will be coded by this sequence of nucleotides? So how many total? You can just count them all or just do like this. 1, 2, 3, 4, 5, and 6. So the answer is 6. Write down the sequence of amino acids. So you need to look for the sequence that this section of mRNA codes for only. Okay, here is something that's very important. Only when it is started with a start codon. Okay, so you know that start codon is AUG. So what we will do, we will look into the segment as AUG. So all the A's you need to check. This is A, but this is not AUG. So cancel it. Go further. This is A and this is AUG. So no need to say what is this. No need to say what is this. Only what you will do, you will start from AUG. Fine. So write down the sequence of amino acids. So AUG, we know that this is meth. Okay. After that AGU. So we will go up. AGU, so there is AGU, this is SER. Okay, SER. Next one, this is done, this is done. This is better how you do it, put ticks next to them. ACA and CGC. Okay, ACA is THR and CGC. So CGC ARG. So THR and ARG. So THR ARG. See, this is what you need to learn. How many total can be coded? Total. How many total can be coded? There were 6. But what if you start with the start codon? There are only 4 that can be coded. So there can be difference. Okay. Use the genetic codon table below, not below actually, this is above. to identify the amino acid X, Y, and Z. So how you do the X, Y, and Z over here? So this is the codon, that is X. This is the codon, that is Y, and this is the codon, that is Z. You can name them as per the table. AUG, of course, this will be meth, and CGA, and the next one we can name them. I believe this is CGA is alanine, ALA, alanine, sorry, and GUU is through valine. Okay, so you can write them here. This is ALA, and then this is MET, and then this is VL. I told you, if the table has the full names, please write full names, don't write abbreviations. Okay, complete the table below by writing the sequence of the pre-mRNA that will be transcribed from the provided DNA sequence of the amino acids, and the amino acids that will be coded by this mRNA after processing, mean after splicing. The provided genetic code chart will help you to answer. DNA template is over here. We need to now sequence the pre-mRNA, mean code everything down. So if it is T, of course, going to be A, thymine with adenine, A with T, but since we are writing mRNA, we cannot write T, so we will write U, and C with G, so it is A, U, G, and so on. You can say C, A, U, then G, A, and then U, G, C. Amino acid sequence formed after splicing. So what you have to know over here, since these are introns, so after splicing we know that introns removed. So this one and this one will not be part of the amino acid which will be made after splicing. So we will take only this and this. So AUG is very famous, meth, and UGC we need to go up to find UGC. So it's here, that is cysteine. Okay, so meth and cis, that's it. With that, we reach to end of this topic. See you again in the coming topics. Thank you.